Students, today we are going to study in Science-2, Chapter 2, Life Processes in Living Organisms, Part 1. Today we are going to see the third part of this topic. Cell division is one of the very important properties of cells and living organisms. Due to this property only, a new organism is formed from existing one. We had studied in the last part about mitosis. Today we are going to study about the cell division meiosis. Meiosis occurs in germ cells. There are two stages of meiosis, meiosis 1 and meiosis 2. By meiosis, from one diploid cell, 4 haploid cells are formed. Meiosis occurs in germ cells. Before the cell goes through meiosis stage, there is a preparatory phase called as interphase. Here the cells goes through 3 phases, namely G1, S and G2 phase, in order to prepare for actual cell division. What happens in this phase is the genetic material replicates. The DNA is replicated to make identical copy of each chromosome. Other organelles of the cells also get duplicated in this phase. Among duplication of all organelles, centrosome duplication is very important as it contains centrioles which forms the spindle fibers needed to separate sister chromatids. Now it is set to move and enter the meiosis stage. Meiosis is carried out in 2 rounds, meiosis 1 and 2. This is to obtain 4 daughter cells, each with half the number of chromosomes compared to the single parent cells. Meiosis 1, wherein the homologous chromosomes undergo crossing over and hence there is genetic recombination. The homologous chromosomes are divided into 2 groups and from this 2 haploid cells are formed. Prophase 1 of meiosis is lengthy phase which is divided into 5 phases: leptotin, zygotin, packetin, diplotin and diakinesis. During packetin, crossing over takes place. In leptotin stage, initially the chromosomes start condensation and they become compact during leptotin. In zygotin, homologous chromosomes starts pairing and this pairing is called synapsis. The structure called synaptonemal complex develops to hold chromosomes in place during this pairing, and each chromosome's chromatid arms and form structure called bivalent or tetrad. In packetin, crossing over of non-sister chromatids of homologous chromosomes takes place. Genetic recombination is produced due to such exchange. The homologous chromosomes still remain paired together at the sites of crossing over. In diplotin, synaptonemal complex dissolves and the homologous chromosomes of the bivalent separate except at the point of crossing over. Thus, it looks like X-shaped structure called the chiasmata, as you can see in the slide. The last phase of prophase is for termination of chiasmata. The spindle fibers originate and the crossing over homologous chromosomes are now separated. The nucleolus disappears and the nuclear envelope breaks down. Next phase is metaphase 1. The homologous chromosomes arrange on the equator with random orientation forming metaphase plate. The next stage is Anaphase 1, wherein the homologous chromosomes separate from each other and move towards the opposite poles. Finally, cell enters telophase along with cytokinesis 1, wherein two new cells that are formed is haploid daughter cell. Now these two stages, namely telophase and cytokinesis, helps in the formation of nuclear membrane surrounding the chromosome and the nucleolus. The daughter cells are haploid, which are formed from the diploid parent cells. You can see that chromosomes are still attached to sister chromatids. In order to separate the sister chromatids from each other, each haploid cell undergoes another round of division. Before entering, before moving ahead, that's meiosis 2, the haploid cell enters a small resting phase called interkinesis with no activities, which is also called as a resting phase. Now it is set to move on to the next meiotic division called meiosis 2. It is also called as mitosis of haploid cells. Now it has four stages: prophase 2. You can see that nuclear membrane and nucleolus starts disappearing, chromosomes start condensing and become short and compact. The next phase is Metaphase 2, wherein microtubules of spindle interacts with the chromosome. This causes chromosomes to move and align along the middle of cell, that is at the equator plate. Sister chromatids are held at centromere, confirming the presence of protein structure called kinetochore at the surface. Kinetochore holds chromosome and attaches them to the spindle. The next phase is Anaphase 2, wherein you can see that the sister chromatids separate at the kinetochore's daughter chromosomes along with divided centromere move towards the opposite poles as microtubules shortens. The poles of the spindle fiber also move apart and also help to separate the chromosomes. As chromosomes separate completely into two sets, one, you can see that the two cells have simultaneously undergone meiosis 2, so we get four haploid cells at the end and are called gametes. So in meiosis, crossing over between non-sister chromatids of homologous chromosomes occur, and this results in genetic recombination. So the four daughter cells formed after completion of meiosis are genetically recombined and not exactly alike, and also not exactly similar to the parent cell. Spores and gametes are formed by meiosis. Meiosis helps to restore and maintain the chromosome number.